What you doing?
Oh, I'm looking for some nodules. It's pretty fixating.
I think you're talking about nitrogen fixation.
Hi, I'm Andrea Reynolds, and I'm going to be talking about the biological significance of nodulation. Nodulation is a component of the symbiotic relationship between legumes and gram-negative soil bacteria, which can fix nitrogen. The first biological significance is the symbiotic relationship between bacteria and legumes. First, we must understand how nodulation of the legume root produces nitrogen through symbiosis. Soil bacteria invade the roots of legumes and produce nodules where the bacteria can be housed. The soil bacteria, which are prokaryotes, are the only organisms that can fix nitrogen, while eukaryotes do not have the enzymes to do so. The enzyme nitrogenase can convert atmospheric gaseous nitrogen to fixed nitrogen. This symbiotic relationship maximizes the process of nitrogen fixation for the plant's benefit, as well as providing the bacteria with carbon. Next, I will talk about the significance of nodulation to the global nitrogen cycle. Bacteria within the nodulations of legume roots are known to make the largest contribution to biological nitrogen fixation. This is important because the increase in human activities have altered the nitrogen cycle mainly through industry and fertilizers. Through nitrogen fixation of atmospheric nitrogen, bacteria in nodulations fix nitrogen to usable forms for plant growth. Nodulation is important for fixing nitrogen to combat the increase of nitrogen in our atmosphere. The third significance has to do with low nitrogen areas. Nitrogen in many processes is a limiting nutrient. A limiting nutrient is a nutrient that limits the growth of an organism when in short supply. Nodulated legumes can grow in soil that are in low nitrogen, where other plants cannot grow because of the availability of fixed nitrogen. Plants with nodulations infer an advantage over plants without nodulations in environmental conditions that have low nitrogen. The last point of biological significance is the sustainable production due to crop cover. Crop covers are of high concern in agriculture due to chemical input, improving soil quality and crop yield. Fertilizers enriched with nitrogen have been used to yield crops. However, fertilizers have been shown in the recent years to have extreme pollution effects on groundwater, so farmers seek to reduce chemical use, improve soil quality, and still have successful crops. Here you can see a crop cover of legumes with nodulations, opposed to a crop cover without nodulations. Legume crop cover is planted in soil to add nitrogen. A substantial amount of nitrogen is produced. It later degrades and decomposes to leave the topsoil enriched with mineral nitrogen that plants can use.
Hi, I'm Randi, and I'm going to be talking about the developmental process of nodulation.
The way that nodulation develops, plant roots release these chemical signals called flavonoids to attract rhizobia bacteria in times of nitrogen depletion. Rhizobia are then drawn to the root and attached to root hairs, where they then emit signals called nodulation factors. The root hair responds to these nodulation factors by curling up, as you can see here, and encompassing the bacteria. This way it brings the bacteria closer to the root cortex here. The rhizobia induce formation of this thing called the infection thread. They enter the root through this infection thread, which contains reproducing bacteria. This allows them to travel to the cortical cells, so that's right here, and pericycle cells here, where they signal cell division via those nodulation factors I mentioned earlier. This produces a nodule primordium, which you can see here. This is what an actual infected root hair looks like. You can clearly see the infection thread and bacteria dyed blue, and see how the infection thread leads from the curled root hair into the cortex. This is also a good example of how the infection thread branches off to infect more cells in the cortex. Bacteria are then released from the infection thread into the host cells, but they remain surrounded by a peribactoid membrane. If this membrane doesn't form, the host can recognize the bacteria as an infection and signal its defenses. If the bacteria isn't protected, it can lead to ineffective nodules. The infected cortex and pericycle cells continue to divide until they eventually fuse, forming the nodule. Once the nodule is formed, the bacteria change to bacterioids and begin fixing nitrogen. Nom 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 nom. The nodule makes contact with the plant's vascular system to allow absorption of nutrients.
Hi, I'm Taiya, and I'm going to be talking about the mechanisms of nodulation and nitrogen fixation.
Going to start off with nitrogen gas with 8 protons, or H+, 8 electrons, and 16 ATP. And that's going to be converted via nitrogenase into 2 moles of NH3 with hydrogen, 16 ADP with 16 inorganic phosphate molecules. So let's get to know our superstar enzyme nitrogenase. This enzyme is a complex of 2 metalloproteins. The first component is referred to as the iron protein and is the smaller of the 2, so we can picture it looking like this. The second component is the iron molybdenum protein. This is the bigger half of the component, so we can picture it looking like this. Neither of these components is capable of catalyzing the reduction of N2 to NH3 by itself. Both components are also extremely sensitive to O2. In fact, the iron protein is so sensitive to O2 that the component undergoes an irreversible inactivation in the presence of it. The inactivation of component 1 would make nitrogen fixation impossible. This explains why nodulation occurs in the root system versus other places on the plant. This enzyme requires anaerobic conditions in order to be happy. Now we're going to look at the electron exchange that makes nitrogen fixation possible. We're going to start with reduced ferredoxin, which is going to transfer its electron to our iron protein, which is going to start in its oxidized state. But as the electron is added from the ferredoxin, it's going to turn into its reduced state. Simultaneously, ferredoxin is also going to return back to its oxidized state. The reduced iron protein component is going to move the electron to the iron molybdenum protein component. This is going to take that component from its oxidized state into its reduced state, and it's going to return the iron protein component back to its oxidized state. This occurs with the simultaneous hydrolysis of ATP into ADP to use as a power source. The reduced protein transfers the electrons to the molecular nitrogen with 8 protons to get the resulting 2 ammonia and 1 hydrogen molecule. The protein is also returned back into its oxidized state. Overall, this is going to be the pathway of our electron as it moves through the system. ATP hydrolysis comes in because the activation barrier for the breaking of nitrogen triple bonds is enormous. However, the reduction of molecular nitrogen to ammonia is thermodynamically favored and therefore stable once it is reached.